Today we'll be presenting our project, which is Mitosis Model and Karyotype Cancer. Essentially, the main purpose of our project is to understand the biological process of mitosis and how it relates to the development of cancer in an organism.
One of the main ideas of this project is the compartmentalization of genetic material. As you can see in the image, the string, which is meant to represent DNA, takes up a lot of space when stretched out to its maximum size. However, when the DNA is compacted, the same amount of DNA can fit into a much smaller space. The compaction and compartmentalization of DNA is applied during mitosis in order to divide genetic material along two new daughter cells effectively.
In this slide, we see the cell during interphase right before mitosis begins. In this model, the green pipe cleaners represent the cell membrane, the pink pipe cleaner represents the nuclear membrane, and the purple yarn represents the genetic material in its loose form of chromatin. In this phase, the cell is ready for cellular division.
In this slide, the first step of mitosis begins, prophase. Essentially, as the model shows, the genetic material organizes itself from chromatin to a more compacted form of DNA, or chromosomes. The nuclear membrane starts to dissolve as well.
In this slide, the next phase of mitosis is shown, metaphase. In metaphase, the chromosomes align along the metaphase plate, which is an imaginary line along the middle of the nuclear zone. The spindle fibers, which are represented by the clay and the white lines, start to attach to the centromeres of the chromosomes, which are represented by the blue beads.
In the next phase of mitosis, or anaphase, the spindle fibers attach to the centromeres, or the chromosomes, and pull the chromatids to the opposite poles of the nuclear zone. The remnants of the nuclear membrane are still present, which are represented by the pink pipe cleaners.
In the final stage of mitosis, or telophase, two new nuclear membranes form on opposite sides of the cells, each with an equal number of genetic material in each as seen in the model. Pressure starts to form on both sides of the two poles of the cell membrane, leading to the finishing touches of cellular division.
In this stage, cytokinesis occurs and the cell membrane is effectively divided into two new separate cells. As seen in the model, each cell is about the same size and the same amount of genetic material.
This slide shows the two new identical cells that are a result of the meiotic process. As shown in the image, the two cells are the same size as the original cell or the parent cell. These cells are ready to grow and eventually divide into more cells via mitosis as well. We are able to apply the principles of this project to both ELA and social studies. In terms of ELA, we were able to make journal entries recording the project process and our experiences with project-based learning.
In the social studies component, I learned mitosis is an integral part of any biotechnological process and application. The understanding and manipulation of mitosis have been instrumental in advancing biotechnology and its diverse applications. That's all for our video on mitosis. I hope you learned something after watching this video. Thank you.